Hello aspirant, welcome to Vision Institute. This is a quick by series, and this quick by series will be discussing on the physiological effect of plant hormones. We know that the growth and development of the plant is regulated by a number of chemicals, and these chemicals are usually produced in small amount. So these chemicals are called as plant hormones, or they're also commonly called as phytohormones. There are 5 plant hormones that have been discovered so far, and these 5 hormones have been classified into 2 types based on their function. We have auxin, gibberling, and cytokinin that are called as plant growth promoters, whereas ethylene and abscisic acid they are also called as growth inhibitors. We will be discussing on the physiological effect of each of these plant hormones. The first is auxin. Auxin it's a plant growth promoter, and it brings about root formation. Auxin promotes root formation. That is why it is used in tissue culture in order to initiate root formation as well as cell division. And not only in tissue culture, it is also used in plant propagation, where they initiate the formation of root in stem cutting. Auxin induces parthenocarpy. Parthenocarpy means the formation of fruit without fertilization. So the application of auxin produces seedless fruit, for example, seedless tomatoes. Auxin also promotes apical dominance. In plants, we know that there are 2 types of bud that is produced. One is the apical buds. These apical buds promotes the elongation or the increase of the height of the stem, whereas the lateral bud increases the length of the stem horizontally. When auxin is applied to it, it promotes the growth of the apical buds, but it terminates the growth of the lateral buds, and that brings about apical dominance. There are some certain auxins that are also used as herbicide, mainly 2,4-D. 2,4-D is a type of auxin. It is used as herbicide and is mainly used in order to kill dicot weeds, and it's used by gardeners in order to keep the garden and lawn free from weeds. Moving on to the next, we have the next plant hormones, gibberling. This gibberling, the main function of physiological effect of gibberling is it brings about elongation of the internodes. Since they bring about the elongation of the internode, they also bring about the increase in the length of the stem. And this gibberling is used in grapes and sugarcane cultivation, where it helps in increasing the stalk of the grapes, whereas in sugarcane growth, when the sugarcane plants are sprayed with gibberling, it helps in increasing the stem of the sugarcane and thereby increasing the yield of the sugarcane. Auxin also delays senescence. If you want to keep your fruit on the tree for a longer period of time, then you can apply gibberling, because gibberling delays senescence and ripening. Gibberling promotes balding. In plants like cabbage and then beetroot, the internodes are greatly reduced. And when the internodes are greatly reduced, the leaf overlaps with each other around the shoot apexes. That is called as rosette appearance. So when you apply gibberling to those rosette plants, it helps in balding. That is, it helps in the elongation of the internodes. Okay. Moving on to the next, we have cytokinin. This cytokinin is also a plant growth promoter, and these cytokinin are usually found in part of the plant where there is rapid cell division taking place. So you will find cytokinin in developing or growing buds, or you can also find it in young fruits. This cytokinin brings about the production of new leaf. It also increases the amount of chloroplast in plant. Even though cytokinin is a plant growth promoter, but they do not promote apical dominance, but instead they initiate the growth of the lateral buds as well as advantageous shoot formation. One key point to be noted here is that cytokinin helps in nutrient metabolism. It helps in the transportation of the nutrient across the plant membrane. Now moving on to the next, we have ethylene. Ethylene is different from the rest of the other phytohormones because they are gaseous in nature. When you have a ripened banana at home, what do you smell? You get a fruity smell. That is the ethylene smell. Okay. So here, this ethylene is chiefly associated with the region of the part where there is synthesis and ripening taking place. So ethylene promotes abscission as well as synthesis. Ethylene is chiefly associated with the ripening of fruit. The most common source of ethylene is a compound called as etyphone. Etyphone is a source of ethylene and is chiefly used to hasten the process of ripening, especially in apples and tomatoes. This ethylene also promotes root formation as well as they also promote root hair formation, and thereby they increases the surface area of absorption in plants. And now moving on to the last, we have abscisic acid. This abscisic acid is a plant growth inhibitor. It inhibits seed germination and thereby it induces seed dormancy, so that the seed can overcome the certain period of time where it is not suitable for them to germinate. This abscisic acid also stimulates the closure of stomata. When the plant is under any stressful condition, this abscisic acid signals the closure of stomata in order to avoid unnecessary loss of water. That is why this abscisic acid is also called as stress hormones. Overall, this abscisic acid acts as an antagonist to gibberling. That is, the function and the physiological effect of abscisic acid is opposite to that of gibberling. Overall, as you can see at the end, all of these plant hormones play a vital role, and each of these together function to bring about the growth, development, and differentiation of the plant. Thank you.